In this video, I am going to introduce researches on biochips and microfluidics for cell handling and analysis conducted at the Nanobio Modeling Lab, Faculty of Science and Engineering, Chuo University. Biochips are devices that allow manipulation and analysis of tiny biological objects, typically having small chambers or grooves on the chip to allow fluid flows. In this example, water flows in a complicated flow paths are controlled by integrated valves to perform sophisticated fluid manipulation similar to an electronic circuit. This kind of devices are called integrated fluid circuits, IFC. The next example, which I conducted at UCLA during my doctoral course, is a biochip that efficiently mixes magnetic beads in a microchannel to selectively capture specific cells. Active research on biochips is being carried out using such microfluidic devices to culture cells that simulate human organs and conduct drug tests. Here, I will discuss single-cell analysis. There are many different types of cells, and it is becoming clear that even within similar cell populations, there are considerable differences among individual cells, similar to the individuality of humans. With conventional methods, many cells are placed in a test tube and measured all at once, so only the average value of the cell population can be determined. Using microfabrication, for example, it becomes possible to create microchambers, or containers, that are about the same size as the cells, making it possible to observe and examine individual cells separately. This picture shows many chambers produced in our lab that are about the same size as the cells, and letting the cell fluid flow through them. If you zoom in, you can see that each hole contains approximately one cell. Then, when you run a reagent to examine the state of the cells and cover it with oil, you can see that each cell responds differently. Next, I introduce the open microchannel that we developed recently. Typically microchannels have a closed ceiling, and fluid flows are created by applying pressure via the connected tubings. However, with this system, even if cells are precisely arranged in the device, they cannot be taken out from there. In the open microchannel system, grooves are created on the surface of the substrate to serve as channels. The grooves are treated to be hydrophilic, and water flows through the channels due to capillary action. There is no lid in this system, allowing access from the outside. We created a complex network of these open channels as seen in this electron micrograph. Just a drop of liquid containing particles or cells placed in this channel creates a spontaneous capillary flow that traps the particles or cells in specific locations. After that, you can carefully examine the cells and pick up the selected cells. The thousands or tens of thousands of particles and cells can be trapped in this system to increase the number of cells to be examined. We aim to apply this system to improve the efficiency of cell screening. Next, I discuss another powerful single-cell analysis technique, droplets. Water-containing cells and oil are pumped into this flow-focusing microchannel. Within the channel, the water containing the cells is surrounded by oil and breaks off, forming uniform droplets. If you dilute and adjust the cell concentration, exactly single cells can be encapsulated into single droplets. In joint research with Tokyo University of Marine Science and Technology, we encapsulated hemocytes of Kuroma shrimp into droplets and investigated gene expression in individual cells. As a result, for the first time, we succeeded in classifying the blood cells of Kuroma shrimp. Changing the topic again, I introduce a biochip for investigating the function of adherent cells. The cells on the surface of the intestine are called epithelial cells, and they form a sheet-like structure. Adjacent cells adhere together tightly, separating the inside and outside of the body. The conditions under which substances pass through this cell sheet are important for drug absorption, etc. We discovered that when epithelial cells are cultured on a microchamber smaller than the cell sheet covers the chamber space. In this state, as substances diffuse across the cell sheet, they accumulate in the opposite microchambers and can be detected using a microscope. Furthermore, we developed a microdevice that allows cultured epithelial cells to be observed by flipping them 90 degrees, and succeeded in obtaining very fine cross-sectional images of cell sheets. By combining this with the above permeability measurement technology, it will be possible to investigate how cell structure and function are related. Finally, I discussed the vibration-induced flow for manipulating fluids, cells, and molecules. This is a joint research with Professor Takashi Hayakawa from the same department. First, we fabricated micropillars like this one, placed water over them, and applied small vibrations. This creates a net flow around the pillar, called vibration-induced flow. This flow can be used to mix small amounts of fluid efficiently, or to detect molecules and nanoparticles for disease diagnosis by inducing particle aggregate formation in fluids at certain locations. Thank you for watching the research introduction of the Nanobio Modeling Lab, Faculty of Science and Engineering, Chuo University. If you are interested, please join us in research and enjoy science and engineering together.